Hello students, how are you all? I'm sure you are all doing well. Before we begin the class for today, children, let me give you a little bit introduction about this app. You can look onto the screen. The name of the app is NEET Biology at Locus. So it's a learning app, especially teaching you and coaching you on biology subject, which is the main subject in NEET. So this app is for all the students who want to get through NEET this year, that is 2023, and the coming years. So if you want to know more about the app, children, I have provided a link in the description box. Please click onto it, download the app, and we will get in touch with you. Then if you like my videos, please watch all my videos till the end. And if you like them, please subscribe and hit the bell icon. So without wasting time, let's get into a very interesting topic that I am going to teach you today, and that is from Unit 6, Principles of Inheritance and Variation, and the topic is linkage and recombination, which I believe is an exciting topic. So, what points are we going to learn in this class today, children? We are going to learn about linked genes, linkage groups, Morgan's experiment on linked genes, types of linkage, crossing over, recombination, and the significance of crossing over and recombination. Then previous years need questions related to the topic. Let me tell you, children, in all of my videos, I am going to give you, at the end of the video, I'm going to give you a set of questions which were asked in previous NEET, so that you would know what kind of questions come to NEET. I'm sure many students would be having this doubt, like how would be the question type, like what is the difficulty level, and how are the questions formed. So all those doubts would be cleared if you go through those questions. And then I am also going to share what would be the weightage for each of the topic. Now, if you see, children, this topic is very important, and you can expect somewhere around 10 questions coming from this topic. So then let's get into the introduction. Now here, if you look onto the screen, this is all about genes we are talking about. Now I'm sure you all know about what these genes are. I'm talking about the biological genes, and for all those who want to know what genes are, so genes are considered as the unit of inheritance. These are the factors which get transferred from one generation to the other generation. Means genes carry characters, and these characters, they move on from parent to offspring. So whatever characters parents are having, they get carried to the next generation. So those are genes. And technically, what are genes? Genes are, you can say, they are a sequence of nucleotides which are present on the chromosome. And where are chromosomes located? Obviously, they are located inside the genetic material, right? Or inside the nucleus. Now, today, what are we going to learn is, if you can see the first point, the number of genes in an organism far exceeds the number of chromosomes. That means what? We have more number of genes and very less number of chromosomes. Now, for example, you take human beings. How many chromosomes we have? We have 46 or 23 pairs of chromosomes. So, though we have 23 pairs of 46 chromosomes, we don't have one gene on one chromosome. Actually, it's not like that. There are more number of chromosomes and all these, I'm sorry, more number of genes. And all these genes are just, compactly arranged, or you can say they are squashed uh, on one chromosome. So, a single chromosome bears several genes. I have given few examples, like you can see, Daphnia pulex. Now, what is Daphnia pulex? It's a arthropod. It's an animal, and it has 31,000 genes. So just imagine this arthropod, right, is a very small animal. I'll actually show you the picture after this slide. It is having 31,000 genes. Actually, it has more number of genes, more than we have. If you see, humans are having 23,000 genes, and Daphnia is having 31,000 genes. And we carry genes close to number to mice and roundworms. I'm sure you don't like comparing yourself with a mice and roundworm. But the point is, see, you cannot compare the body of an organism with that of, or the size of the organism with the number of genes. Daphnia, being so small, has 31,000, and human beings have 23,000. Then look at the small plant. Even the small plant shares the same number of chromosomes, which is a flowering plant, and it's called Arabidopsis thaliana. Fruit fly has 14,000 genes. I'm going to talk about fruit fly in the coming slides. Then yeast, which is a fungi, it has 6,000. E. coli, which is a bacteria, it has 3,200, and zebrafish, it has 5,000 genes. So the point is, there are more genes and less chromosome. Now, if you look onto the screen, children, this is Daphnia pulex, very sweet, or you can say a cute animal. And this is the flowering plant I was talking about, and that's fungi, that's E. coli, and that's zebrafish. Let's get into linkage. Now, what is linkage? Linkage, it's a phenomenon of genic inheritance in which genes present on a particular chromosome show the tendency to inherit together is known as linkage. Now, for all those who can't get this definition, let me give you a simple explanation of what linkage is. Now, first of all, it's related to genes. Linkage is found in certain kind of genes called as linked genes. Now, in spite of calling them linked genes, I would call them as the best friend genes. Now, why best friend genes? These are certain genes in the body, in our body or in other organisms' body, they always get inherited together from one generation to other generation. Now usually what happens, genes they segregate independently, but these genes do not segregate independently, but they get inherited from one generation to the other, from parent to offspring as it is. So I am calling them as best friend. Like how best friends, what do we do when we have a best friend? We always like to go for a movie together. We want to share our food together. We want to study together. And then we also, share our secrets with our friends. So we are always together. So that's why I'm calling these genes as linked genes because, or best friend genes, because they get inherited together from one generation to the other generation. Now, if you look onto the definition here, these genes are present on a particular chromosome or a particular chromosome and they are very close to each other. When I say close to each other, they are physically very close to each other and they tend to get inherited together from one generation to the other. Now, such genes retain their parental combination. Now, what do you mean by parental combination? Suppose there are two genes A and B, which are very close and which are linked. A and B, whatever characters they are carrying, the same characters, it get inherited from one generation to the other as it is. So, whatever parent combination is, the same combination you would see in the offspring also. How are genes arranged, children? Genes are always arranged in a linear manner. Now, for all those who don't know, let me tell you this is how the genes are arranged. Now, one example I've given you in the form of a picture. Here you can see onto the screen, please follow my cursor. Now, this is one chromosome. On this chromosome, you would see three genes A, B, and C. Now, these genes, if they are linked, you would see them being pushed to the next generation as it is. But basically, understand that genes are arranged in a linear manner on the chromosome. Now comes to the point, how much linkage? How do we calculate linkage? Or you would uh, call it calculating. In spite of calculating, you can use the word strength. Now, the strength of linkage depends upon obviously the distance between them. The closer these genes are, the more linkage is, or more strength of linkage is, or more percentage of linkage is. So, you can look onto the sentence here. The strength of linkage between two genes is inversely proportional to the distance between them. Now, let's get into the people who worked beyond or behind this discovery. And you might have heard about this scientist. He is a very famous scientist and his name is T.H. Morgan. T.H. Morgan and Castle in the year 1911, they were trying to find out what is this linkage all about. So they were the first people to discover and describe this linkage and linked genes. Now, the discovery did not start in 1911, it started somewhere in 1908. And on which organism they were working on? They were working on Drosophila or the fruit fly, which is like a very common fruit fly or a fly that you see, always around vegetables and fruits. Now, the question is, why did Morgan and Castle choose Drosophila instead of some other animal? Like they might have chosen a mice or a monkey or some other animal, but there was a reason why they've chosen fruit fly. And for all those who know the answer, I know what your answer would be. The answer is, first of all, they are very small. You can capture them and you can start breeding them in a very little space. So Morgan had his own lab. In his own lab, somewhere in the corner of his lab, he started growing them. And where did he grow? He had these small glass bottles. In that glass bottle, he provided the food medium or the nutrient medium and started growing these Drosophila in a small jar. And within less time, you would see many generations of Drosophila. So you can breed them in less time and less space. So then what happened? He was doing his research on Drosophila and then he found out about these strange genes or best friend genes. Let me also add one point here, children. See, Morgan, during his distinguished career, he wrote 22 books and 370 scientific papers, which is like, wow, who writes 22 books for us? Preparing for NEET or studying 4 or 5 books itself is so hard, and this person wrote 22 books and 370 scientific papers. So let's get into Morgan's experiment and let's find out how did he come to this conclusion. Now, what did Morgan do? Morgan carried out several dihybrid crosses in Drosophila to study the genes that are sex-linked. Now, one more thing, children, Morgan also found out at the end of the experiment that the genes were sex-linked. Now, for all those who don't know what are sex-linked, you can look onto the screen here. Genes, basically, which are present on the sex chromosome. Now, when you talk about humans, we have one pair of sex chromosome. That means females would have XX and males would have X and Y. So if genes are present on one of the sex chromosome, you would call them as sex linked genes, and the inheritance pattern also depends upon how this sex chromosome gets inherited into the next generation. Now, for all those who don't know what is a dihybrid cross, let me give you an introduction about this dihybrid cross. See, di means two. So, when you are considering or when you are trying to cross two organisms carrying two characters or trait, you call it as a dihybrid cross. Obviously, when you are doing a cross, it must be between two organisms, one male and one female. When you consider one character, you call it monohybrid. When you are considering two characters, you call it as dihybrid. So you can look at the definition here, children. A dihybrid cross is a cross between two organisms with both being heterozygous for two different traits. So two different trait means it's a character. I have given one example here. Let's say these are the two characters. So one character is the seed shape, another character is the seed color. So when I am considering two characters and when I am crossing between two organisms carrying those two characters, I will call it as a dihybrid cross. Then also another point is the second example, a cross between round yellow and wrinkled green seeds. Now look at the nature of these seeds, children. See, round and wrinkled. Now this is, we are talking about the seed shape. Now the seed shape is having two variants. Some are round, some are wrinkled. So that means in a particular plant population, some plants are producing round seeds and some are producing wrinkled. Then look at the seed color. Some seeds are yellow and some seeds are green. So what am I doing? I am crossing between a plant carrying this character round and yellow, two characters, two variants, and wrinkled and green, two characters, two variants. So that's a dihybrid cross. So Morgan did the same thing. He didn't cross plants, but instead he took the fruit fly. So what are the two characters here? See, yellow bodied, white eyed female. And what is the genotype? YW. Then with what is the other character? Brown bodied red eye males. So males were brown bodied red eye and females were yellow bodied white eye. Look at the genotype, everyone. This is YW and this is Y plus W plus. Always remember, plus sign is given for wild type. What is a wild type? A particular character or a trait that you see most common, or a common character that you see in a particular population, you call it as wild type. A wild type can be dominant or recessive. So here in Drosophila, brown bodied red eye were very common and yellow bodied white eye were very uncommon. Like, when Morgan started breeding many generations. In one of the generation, he found this mutant type. So you use the word mutant when it is rare. So he took the parents, then he got F1s, he intercrossed the F1s, right? Now look onto the screen here, children. I've taken the same picture which is there in the NCRT. See, the parents are here. You can see this is the female parent. The female parent is yellow and white, that is Y and W. Both on the X chromosome, you have the same genes, and then on the wild type, you have Y plus W plus, and that's the Y chromosome. Then in F1 generation, what did he pick up in F1 generation? He picked up one female, which is wild type, and one male with yellow and white. Now here, if you look at the wild type children, the wild type one on one X chromosome, there is YW, on the other X chromosome, that is Y plus W plus. But still, it's a wild type, right? Now, when he crossed, what did he observe? Now, he observed that in F2 generation, the ratio deviated from 9 is to 3 is to 3 is to 1. Now, for all those who know Mendel's laws, you know that Mendel's one of the law is independent assortment, law of independent assortment, where the genes, they segregate separately, independently, and you find new combinations in the next generation forming, and the ratio would be in the form of 9 is to 3 is to 3 is to 1. But that kind of ratio he didn't observe. Now, for all those who don't know what is this ratio, let me tell you. Suppose if you have 16 plants, in F2 you got 16 plants, 9 plants would be carrying dominant character, you can follow my cursor, 1 plant would be recessive, and 3 is to 3 is the new recombinants formed. But this was not the ratio what he got. Why did the ratio deviate? The reason behind is linkage. Now, how that it happened, let me tell you in a while. Now, for all those who don't know, what is this independent assortment and recombinants and all, this is one picture I've given children. You can see these are homologous pair of chromosome, right? That is uh, on one chromosome you can see capital A, capital B, and on another homologous pair you see its variant or allele small a and small b. Now, during crossing over, what happens is a part of the chromosome gets exchanged. You can see here a part is getting exchanged, and because of this exchange, you see new combination or recombinant formed. So you can see how recombinant is formed, capital A and small a and capital B. So this recombination happens when genes get independently assorted. But because of linkage, you can see the proportion of parental gene combination were much higher than non-parental. That means what? Since these genes did not segregate, they were carried as it is into the next generation. So parental combinations were more, and non-parental here means the recombinants. Recombinants were less. You can also look onto the screen here the percentage, children. Parental gene combination. What are the parental gene combination? You can look here. The parents genotype was 98.7 because they were heavily linked. And then non-parental are the Y plus W and YW plus were just 1.3%. That means only few showed the recombination. Now, if you ask me, children, what is the percentage if the genes are not linked? Look onto the cross B, children, onto the screen. I've also taken it from NCRT, the same example. Here are two genes which are responsible for white miniature and wild type. So you can see here this is the female and this is the male, and when they were crossed, we get the F1s. And in the F2, look at the percentage. I want you to concentrate on the percentage. Since those genes are far away, independent assortment happened, crossing over happened, and recombinants were formed, but there was greater percentage. So you can see parentals were 62.8, recombinants were 37.2. That means what? Because they are not linked, recombinations showed more percentage. Right. And then another point what Morgan observed is the genes were located on the X chromosome. Now Morgan gave two different terms. He gave the term linkage and recombination. So what is this linkage? The genes which showed closer physical association, he called them linkage, And who are recombinants? The non-parental gene combinations or recombinations were called, or he called them as recombination or recombinants. Now they showed some genes are tightly linked, showed low recombination. So always remember, children, if the genes are tightly linked, they would show very less recombination. If the genes are loosely linked, they would show higher recombination. Like the second example I was talking about. So what is the second example here? The white and the miniature wing type flies, they show 37.2, and in the first cross, what we've learned, only 1.3. So that is all about linkage, children. Don't worry if you don't know the terms like, dihybrid, monohybrid cross, or if you don't know how to do this crossing and all, don't worry. I'm going to do some more videos so that you would have a clarity on it. Now let's look into the questions which came regarding to the chapter. This was asked in 2016, yes. In a test cross involving F1 dibreed flies, more parental type offsprings were produced. So when you see more parental type and less recombinants, what will it indicate? You can look onto the option here. The two genes are linked and present on the same chromosome. Simple. Next question. Which of the following statement is not true of two genes that show 50% recombination frequency? So the genes are tightly linked. So if two genes that show 50% recombination frequency, then the genes are tightly linked. Okay. So these are the questions, children. Thank you.